And welcome to this week's Iowa Learning Farms Conservation webinar. And welcome to the last webinar of 2024. Always the highlight of my week when I get to moderate one of the webinars, and definitely that's the case today. I should have put up a slide for a poll for our attendees, how many webinars they've attended this year. I think I'd be at least over 25 that I've got to moderate. So I'm Matt Helmers with Iowa State University and the Iowa Nutrient Research Center, and faculty affiliate with Iowa Learning Farms. Really fortunate to get to help host and moderate the webinar today, and super important topic, and one we get a lot of questions about related to soil health. So we're really fortunate today to have DeAnn Presley, that is a professor of agronomy at Kansas State University, here today to talk to us about on-farm soil health assessment results, lessons learned. As always, if you have questions, please type those into the chat box, and we'll get those to Dr. Presley at the end of the talk. And with that, the virtual floor is all yours.
All right, thank you very much. So as he said, I'm DeAnn Presley. And in case you didn't know this, I am from Iowa originally. So I got my bachelor's, and I didn't put the year up there intentionally, but I got my bachelor's from agronomy in Iowa State. And then about the end of grad school, or about the end of undergrad, I was like, hey, I'm not done learning yet. So I ended up at graduate school at Kansas State. And I also did my PhD there as well, all in agronomy, but really I'm a soils person. And I am an extension specialist and professor, and I could talk forever. So instead of getting all sidetracked, I'm going to keep going. So I put up a picture of a Milo pile. So in Kansas, when people ask me, how different is Iowa from Kansas? I say, well, it's pretty darn similar, except it's hotter, it's windier, and we grow a few different things. So this is grain sorghum. Today, the research I'm going to be talking about has a lot of co-authors and sponsors and things like that. So when I say we, I'm talking about a lot of people. Peter Tomlinson is a coworker of mine, and he's my partner in crime. And Adam Petty is a master's student who is now NRCS soil scientist for Iowa in Sheraton. And sorry, he's a soil conservationist. And then a lot of more people too. And these are some of the sponsors of our work. So the objectives of our work have been to measure properties of soil health on farms owned by real people. So I've done a lot of small plot research over the years. I still do, but I prefer doing work on farms. I just think it's more fun. And I also then want to get that information back to those people who I've worked with. And that is something that I'm continuing to do all the time. This is a picture of a pencil, obviously, but I've got some platy soil structure here at the surface. And this is my partner in crime, Peter, and this is a farmer, and this is his home place back here. But we were out there pulling some soil cores just last week, finished up all of our soil sampling. This is the last field I soil sampled last week. And so I included this picture because many times our farmers, if they're not busy, which I know many of you are extremely busy people, but if they aren't busy, I always invite them to stop out at some point to see what we're doing. So here what we're doing is using this Giddings on a truck and pulling soil cores to a meter, which is 39 inches. And can't have a soil health talk without a turnip or a radish, right? But also, this is a crazy thing that I saw last week. We were pulling soil samples in this field, and I pulled the image up on Google Maps because I always, like to double check it. And there was an airplane in that field, and for a second, it broke my brain thinking that there was actually an airplane in that field, and then I saw the contrails. So just had to share that. So the research I'm going to talk about today is from samples we pulled in 2022 in central Kansas. So Kansas, here's two counties, McPherson and Harvey. So when you think of Kansas, you might be thinking of wheat, and we do have a lot of wheat. So eastern Kansas is mostly corn and soybeans, especially northeast Kansas. Then you have this belt here. This is where I live. This belt is called the Flint Hills, and it's very rocky, shallow soils. And so we have a lot of pasture. Then you move into central Kansas, where there's a lot of wheat and sorghum historically, but that has shifted to corn and soybeans since I've been working here. And then western Kansas, this is where it's either irrigated or not. If it's irrigated, there's a lot of continuous corn. And if it's not irrigated, it's a lot of wheat fallow or wheat milo fallow, wheat grain sorghum fallow. So in our work in 2022, focused in central Kansas, 2023 sampled in northeast Kansas up in this area where I live. And 2024, I just spent the past several weeks soil sampling out in this area. And then in 2025, we're going to move closer to north central Kansas. And for the other parts of the state, I'll get there eventually. And we have funding to do work in southeast Kansas as well. The soils of Kansas are very diverse. They differ in all those different regions because of parent material and also because of climate. And so I should go back to this image here. So this image is showing this is the two counties, McPherson and Harvey. So this black sort of squiggly stuff, this is a tillage survey that has been done. It goes back to the 1980s, and then we've used actually the same routes from the 80s. It started in 1989. We've been using the same route because we actually had access to those maps in a drawer. And so we've been following those same things to see how tillage has changed over time. So McPherson County, this is the town of McPherson. It's kind of an oil refinery town, actually. There's a lot of oil wells in this area, oil and natural gas. And so you can see that as the survey goes around, we actually ignore this part of the county because there is shallow sandstone outcropping there, and you can see the pastures. And so what we've learned by doing this is this particular county is about 30% no-till, about 6% strip-till, 4% reduced till, and still about 50% CT, which is conventional till in this case. So conventional tillage. And so by conventional tillage, what most people in this part of the state are going to do, if they are conventional tillage, is they're growing continuous wheat, nothing after the wheat in the summer. Then they just do a bunch of tillage and then they plant the wheat again. So it's a really different system from the others. And so our strategy for sampling is these are fields along that tillage survey. And so basically we asked for permission from farmers along the survey could we go in and soil sample? And so we've sampled irrigated, we've sampled dryland, we've sampled pasture, native remnants of prairie, and we've got different crop rotations. Some of these have livestock, some of them have cover crops and things like that. So this is some of the data from the tillage survey. And what we've really seen is it's just interesting how reduced tillage just doesn't have much of a presence. It used to early on, and it's dropped. And so conventional till's kind of stayed conventional till. No-till has taken up all of the reduced till. And the other thing we see is that if people are the most often no-tilled crop, this is stacked here. So soybeans is this bar here. That would be the most commonly no-tilled crop. And the most commonly tilled crop would be wheat. So yeah, so this is some work I do. I'm working with NASA. NASA has two different projects. One is called Harvest and one is called Acres. And within that, there's a lot more projects. But the Acres project is really focused on looking at United States and agriculture and helping farmers with a lot of different things. But in this case, the person I'm working with in this on the left side, you can see talks about low intensity tillage frequency. So for whatever reason, Harvey County has more no-till or less tillage intensity, and McPherson County has a lot of tillage. And also we found that tillage changes over time. So there was a study published in 2022 that said that no-till lost about 5 million acres between 2012 and 2017. And we know that tillage practices don't necessarily stay the same. So in our particular area, with working with NASA for those two counties, well, actually it's a larger area than that, but 15,000 acres are adopting conservation tillage, such as no-till or strip-till every year. But 10,000 acres are changing back to tillage in any given year. So it's complicated, but our typical pattern in our study area would be 3 years of no-till, followed by intensive tillage, which is usually wheat. So I'm going to get into the systems a little bit more. Again, this is Adam here. We look like we're dressed on safari because it's hot in Kansas in the summer. We were out there this is probably August or September after the wheat came off. So this is a conventionally tilled wheat field, and there's a lot of those where this would be multiple tillage passes. And you can see it's also pretty dry. And this is the surface here, and you can see the surface is falling apart quite a bit, really soft and powdery. We did find some perennial systems, and we clustered the native along with CRP, which I know CRP used to be cropland, and it probably wasn't very good cropland, but we've clustered that for some of our stats that we've done. And this particular farmer is no-tilling. He also has subsurface drip irrigation. He's about the first person to do it in that area. He has 3 different crops he grows in rotation, including soybeans, corn, and wheat. And he also has cover crops in his rotation when possible. So many different systems, many different possibilities. So some of the things I'm going to talk about are soil organic carbon, bulk density. With bulk density, the way we do that, we take one of those long cores and we split it up by depth, and you determine the density, which is mass divided by the dry weight. And we can measure the carbon by sticking it in an oven and basically burning off the carbon. And if you do that, you get the tons of carbon per acre. And of course, with carbon marketing, that's how it's traded. Aggregation is a way of seeing how well the soil holds together when we basically put it in a washing machine. Soil respiration is how much it's breathing, how much the soil is respiring, and that's all. It's really not the roots. It's all of the microbes that come back to life after a 4-day incubation period where they're dried and then wetted, and we see how much carbon dioxide is given off. Active carbon is also called POC, which is permanganate oxidizable carbon. I'm actually not going to talk about that today, but it's considered to be a form of labile carbon. ACE protein is autoclavable citrate extractable protein, which is the readily mineralizable nitrogen in the soil sample. Microbial community structure, which is called phospholipid fatty acid analysis, it tells you who's in the soil, and I've got a lot of data on that I'm going to show you today. And so in 2022, we had 50 fields along our transect, and there are 450 fields per county. So we had about 900 fields to choose from. We did 50, and it took us a lot of time. So I'd love to sample more, but that's how many we got. So we did 2 points per field up to a meter. So this is my dirty lab. So if you're looking at the floor here, this is the dirty lab where we literally wash soil. Water stable aggregation tells us about how well that soil holds together when we submit it to dunking it up and down for 10 minutes in water. And when I explain it to people, I say it's kind of like your grade point average. So the more large particles we have left after we've done that, that tells us so we have different sieve sizes. So a big sieve, middle sieve, bottom sieve. So the more A's on the big sieve you have, the better. And so this graph over here got cut off a little bit. That's in millimeters. And so we have the 0 to 5 centimeter depth, which is the top 2 inches, and then the second 2 inches, and then the rest of the A horizon, or the topsoil after that. And you can see that the gray bar is perennial. So that would be both native and pastures and CRP mixed in that without. And then the blue bar is corn, soybean, and then the C is 3 plus 3 plus crops in the rotation, and D is continuous wheat. And so this didn't actually go, we would have hypothesized that these two would have been switched. 3 crops is better than 2 crops. And sometimes it is in some of the analyses that are later on, but this is science, and it's what really happens sometimes, is that sometimes things don't go the way you think they would. It's different for the bottom of the A horizon. But again, the big difference is between perennial and then these other systems. And so another way to show this is, let's just look at the top 2 inches. And this is Crete, Irwin, and Ladysmith soil series. So these are heavy textured soils like you might find in southern Iowa, like way southern Iowa. Pretty heavy textured, so a silty clay loam at the surface, and then you get into silty clay. And so the perennial has the biggest aggregates, and then again, the 3 crop rotation. And this, when I split them out by the soil series here, again, these two, because they both have B's, aren't different, but this one is numerically a little bigger. And then continuous wheat, again, has the lowest. Those are powdery soils. They just don't hold together when we wash them. So moving into some carbon data. So this is for one soil series, just looking at the Crete. What did we get? So this is the tons of carbon per acre per meter, so down to 39 inches. And I'm going to show you another way of displaying this. This is for all the different soil series, and the Crete on the left and the Cretes on the right. So if we look at all the soil series, or all the soils we sampled, we had as little as 20, which is not a lot, and as much as almost 100. And so for Crete, we ended up with these values of, on the low side, the conventionally tilled continuous wheat was maybe around 50 versus this native was maybe around 75, 70. So there is a spread, but again, it's a pretty big spread, and it's going to depend a lot on parent material. So on the low side, some of these are like alluvial floodplain soils that are sandy and didn't have the thickness of the A horizon. So if somebody asks you how many tons of carbon you have per acre down to a meter, which again is usually how it's going to be traded, this value of somewhere from 50 to 100 or more is probably the ballpark. A lot of the things I'm going to talk about are all about roots and soils. So roots and soils go together. You can see this, I split this core open and you can see these roots just plastered here. This is a clayier soil and you can see they're plastered going right down that crack. So ACE protein is a measure of the mineralizable nitrogen. So protein is really a bunch of amino acids and amino acids is really a bunch of nitrogen. And so here you can see all the different ways that we have this split out, and it's measured in micrograms of protein per dry soil. And perennial again is the highest. I didn't have the stats on this. To be honest with you, I went to run the stats this morning and my stats program needed to be updated as of the end of October. I feel like that happens every time I open that stats program. It's always wanting updated. So let's see, corn and soybean was the second highest, and let's see, continuous wheat was a little higher than I would have expected. But we were sampling these at the end of September and into October, and so the wheat would have been harvested at the beginning of July, so maybe there's just enough roots there that are breaking down to support the microbes. 4-day respiration is, again, the incubation where we put dry soil in a jar, we re-wet it, and then we capture the carbon dioxide being given off, just like if we were having a carbon dioxide sensor measuring my breathing right now. And so, again, you can see sort of this spread. Here, the perennial is actually not one of the higher respirations. It's going to have respiration happening year round, but it won't have like these big up and down pulses like you might have like right after roots are dying from an annual crop. So now let's talk about the who lives in soil. So AMF is our AMF is our vascular mycorrhizal fungi. So AMF grow in long thin strands and they have those relationships. So they're the relationships. The root gives them carbon and then plants give them carbon and in return AMF. There's so much cool work being done on this. They could be providing water, helping to improve drought tolerance, and we can't have big blue stem grasses in the Flint Hills of Kansas without this happening. So the fungal hyphae go and get the phosphorus for the plant, and in return, the plant gives the fungi carbon. They also produce glomalin, which sticks soils together, which goes back to that washing the soil up and down in the washing machine. It sticks the aggregates together, sticks the soil particles together to form aggregates and protect aggregates and make aggregates, and then we get more formation and stability of soil carbon. They are something that people think about a lot, especially if you're into cover cropping, but not every cover crop is going to be they're not all mycorrhizal. So buckwheat radishes and other brassicas like canola, things like that are non-mycorrhizal. So going back to this data, so the perennial had the most, and then corn and soybean on the no-till 3+ crop rotation, they were about equivalent. They both have B's behind them, so statistically they're the same. And then again, the wheat grouped out less. So gram-positive and gram-negative, that goes back to like way back to biology, thinking about whether or not this absorbs violet stain. And so gram-positive and gram-negative, you're gonna see the same trend starting to stack up, where the perennial has more gram-positive and gram-negative, and corn and soybean versus the no-till 3+ crop rotation. And when I say that, no-till 3+ crops, so that would be people that are no-tilling. It would be wheat, and then they might plant soybeans, probably do plant double crop soybeans after that. So wheat, sorghum, or corn, and then soybean would be sort of a pretty typical rotation. So it could be, again, wheat, could be double crop soybean, then sorghum or corn, and then soybean. So that's why I say 3+ crops. It could be actually 4 crops in 3 years as well. So more roots, more support for microbes, and then I think that's why we're measuring higher numbers of those microbes. So the gram-positive are common in the bulk soil and concentrated in the rhizosphere, but they're not as closely, they're right up against the roots as gram-negative bacteria. They are going to be more common in the subsurface soil versus the gram-negative, and again, the gram-positive are larger than gram-negative, and their cells retain the crystal violet stain used in gram staining, back on that last slide. And they're widely dispersed in soil. They decompose organic material that's been partially decomposed by fungi or gram-negative bacteria. And they're resistant to water stress because they can form endospores and then remain dormant for many years. If you get into a drought in Kansas, for example, we have, episodic 1, 2, 3 year droughts pretty frequently here. Gram-negative are a major component of the rhizosphere and improve plant growth by increasing the solubility of many nutrients. So when we're thinking about more microbes and more supporting our biology, gram-negative are really important. And so obviously we know that rhizobium, super important with legumes, fixing nitrogen. In return, the carbon is being given by the plant to the microbe, but it produces ammonium. They dominate surface soils and the rooting soil, and they break down newly added organic matter. And so the next category I want to talk about is eukaryotes. So eukaryotes are these higher life forms, like earthworms, for example. And again, same sort of thing. You can see the mass is going down here. So gram-negative, gram-positive, this mass, I didn't put the units on here, but it's mass per, gram of dry soil or something. And so you can see there's less. It's a food web, just like, we have however many rabbits to support however many coyotes, that kind of food web sort of thing. So this is a PED from a soil that I pulled actually a mile from my house in a project we were doing, same thing, but for our Northeast Kansas soil. So eukaryotes are the fungi, algae, nematodes, earthworms, insects, arthropods, and protozoa that are important in soil ecology. They feed on organic matter, but also on bacteria, plants, and each other. So dog-eat-dog world out there. So after the new residues are added to soil, bacterial populations increase, which then leads to bacteria feeding protozoa, and then who eats protozoa, and from there, the food web. And these guys help mix the soil a lot, and then, of course, create channels for water and air. Fungi are one of the great decomposers, and we see here there's actually no statistical difference between the fungi and any of our other cropping systems, and which I thought was interesting. But they're super important in decomp. They especially the more, the recalcitrant things like lignin. Some of them, of course, pathogens, but many others live mutualistically with plants. Saprophytes break things down. They produce humus, organic matter, and provide plant available nutrients. And they generally prefer more acidic soils than bacteria. And the last thing I'll talk about is actinomycetes. So actinobacteria are gram-positive bacteria that are active in the decomp of organic matter and produce that earthy smell, which is from geosmin. And they can grow deep into the soil. So I didn't want to run too much out of time, so we can go back to any of that if you guys have questions later. But summary point is our conservation-oriented systems, and this is my hand. I picked this little bouquet last week. It was a radish, and you can see a millet of some sort. This tall one cut off the top here, but that was sun hemp, and that was sunflower. So this is a farm where this farmer plants wheat, and after the wheat, he plants cover crops. And in this case, he didn't graze them, which is a pretty rare thing. Most people in Kansas are either after wheat going to plant double crop soybeans and harvest those, or they're going to plant a cover crop or they might plant like sorghum sudan grass or something like that. But usually, if it's planted after wheat, it's either double crop soybeans or it's eaten by an animal. In this case, this one wasn't. It was just a really pretty mix. But in conservation-oriented systems, we generally have more crops in rotation. They're either completely 100% no-till, which is not as common as you'd think. There's a lot of people that do some occasional tillage. I didn't say this earlier on, but Kansas has somewhere in the neighborhood of 11 million acres of no-till every year out of our 20-some million acres, or 23 million acres of cropland. So technically, Kansas has more acres of no-till than anybody else, but that's usually because we're conserving water with that lack of tillage, because again, it's drier here. Cover crops, I think our cover crop adoption rate is somewhere in the 5% range. So there are cover crops, and there's valleys, especially where people have cattle, where you'll see more cover crops. But our conservation-oriented systems were not that much different than like a two-crop system of a corn soybean or a wheat sorghum or something like that. But our 2 and 3 crop rotations were better in most respects. And I say better with air quotes there. Better than most respects than continuous wheat, which again is also conventionally tilled. So and perennial systems of, as we would expect, to have healthy soils, the healthiest soils. But again, I'm not advocating that we put it all back in grassland. I'm just saying it's an interesting thing to measure. So our next steps, I didn't talk much about irrigated versus dry land, but that is going to be an important thing, especially as we move farther west. We're really curious to see what effects that has. Livestock versus none. This is a pretty rare occurrence of seeing sheep, but we went and broke the ice for them to drink water last week. But more commonly we see cattle. So Kansas is we're usually number 3 in cattle, possibly number 2 in cattle some years, but we do have a lot of cattle here. And here's my contact info. I would be happy to email this to anybody if you want to see this. This, again, most of this is written up in Adam's master's thesis, which I need to turn into a publication, and we're working on getting it back to the farmers as well. But Twitter is something I use a lot for work, and I love to talk to people. So, and there's my email address too. So I'll take any questions as soon as the panel is ready to start delivering them.
Okay. Somebody said they're talking about Alberta for beef. So, well, you're across the border there, Jack. Somebody did ask, I don't know if you can do this is to put the link for the master's thesis in the chat box. I don't know.
I absolutely can do that. I'm just going to make it look ugly for just a second.
That's okay.
I'll do that. That's a good one. Let's see, chat. There we go. There we go.
Okay.
There it is. And then I'll go back to show mode.
Okay. All right.
There we go. How's that look? Look through the chat here too.
Okay. Just one second. Okay. Oh, darn it. Oh, I'm struggling now. Okay. Yeah. So let's go to the next slide. And if you are, there we are. If we can go to the next slide, DeAnn.
Oh, your next slide.
Yeah,
Yes.
Yeah. Next slide. Sorry. Okay.
I was snooping through the chat.
Yeah. No, good. You're fine. Yeah, I was trying. Okay. That the thesis is in the chat box now. All right. If you watch this webinar live and are wanting to earn a CCA CEU, please email Elena Whitaker, and her email is alenaw@iastate.edu by 5:00 today. Include your name, the name you entered to watch the webinar, and your CCA number. Again, alenaw@iastate.edu. We can go to the next slide. Okay, one last time for 2024, if you're willing to fill out a voluntary demographic survey, we'd greatly appreciate it. If you filled one out for 2024, thank you very much, and you don't need to do another one. But if this is your first time on one of our webinars and you're willing to fill that out, we'd greatly appreciate it. You can scan the QR code or go to iowalearningfarms.org/survey. Next slide. Okay, and we are back in 2 weeks. Dr. Adam Janke will be joining us. So please tune in 2 weeks, January 8th. That looks like 3 weeks, but okay. Anyway, be back on January 8th. I think that is 3 weeks from now. Dr. Adam Janke will be with us Wednesday, January 8th. Okay, maybe next slide. These webinars are all archived as long as all the recording goes correctly. So go to iowalearningfarms.org/webinar site. And I think there might be one last slide. Yeah. Plug the Regen PGC webinar series coming up starting January 14th. So looking at perennial ground cover. So please tune into that. Those will be on Tuesdays. So Dr. Dan Anderson and Dr. Raj Raman are leading that effort. Also, another webinar that we'll be having tomorrow, or some of us will be involved with, myself will be involved with, partnering or working with the Iowa Nutrient Research and Education Council, talking about some of their survey information. So tune into that. You could Google Iowa Nutrient Research and Education Council and find out information, I think, on their webinar tomorrow. Okay, let's get to some of these questions here. If wheat is in the crop rotation, what crop does it typically follow?
Usually after soybean.
Okay.
So typically it'll go, and this kind of depends. Sometimes people will stack 2 wheats in a row. So they'll do wheat and then either fallow or double crop soybean and then back to wheat if they can. It just kind of depends exactly where they are, north to south and east to west in Kansas.
Yeah.
But yeah, so it's usually wheat, sorghum, soybean, and then back to wheat. And when I say sorghum and milo are going to be sorghum and corn are interchangeable, meaning that most people, if they can grow corn, would rather grow corn, to be honest. But if the ground is a little bit marginal or the precip's a little marginal, then they grow sorghum.
Okay. How much sorghum is still grown or is grown in Kansas?
We're the #1 state for it.
Okay.
So
Okay.
I think 3 million acres or something like that.
Okay.
Something like that. I could be off by a 500,000 or so either
Okay.
direction. But when I first moved here, Kansas had about 12 million acres of wheat. And I think we're down to like 7 point something million acres of wheat. So we lose wheat acres every single year.
Yeah.
We're losing wheat and sorghum acres every year as you know, the drought tolerant sorghum or grains, corn hybrids and wheat. Gosh, I'm got tongue tied. Corn hybrids and soybean varieties become more and more drought tolerant.
Okay. I guess there's kind of a question on this, but how I guess can you relate some of the areas crop yield to any soil health improvements, or are you able to tie that together at all to try to help sell some of these practices that improve soil health from a crop yield standpoint?
There's definitely work happening on that area, and that's actually the power of the relationship with NASA is that when I have these fields that I know that the tillage practices have been this, that, or the other for many, many years now, they can look at that with their NASA tools and say, okay, what's that doing for relative yield differences over time? I would say that I think that the data's a little thin. We're working on it. I think that there's less, and what farmers tell me is that there's less up and down. It's more level. Like I had a farmer, that farmer I showed the picture of, I said to him, I always say to people, oh, so what would be a great yield you've had in this field? And he was like, I don't go for great yield. I go for yield. I
Yeah.
go for yield and profitability. I'm not going for top end yield. I'm going for yield and profitability. And I was like, yes, I need to remember that. So I think it's more about resilience and less crop failures.
Okay. Are there in areas that they're using cover crops, are they concerned about soil moisture in many of those areas?
Yes. So that is our #1 limit to cover crop adoption. So as you take Kansas and just keep stepping across to the west, that's going to be a bigger and bigger and bigger issue. Like literally every county you move west, that'll be the bigger barrier. So we have a small corner of southeast Kansas where we would say we have too much water, but other than that, we always have too little water. And so it's a serious issue. And that's why I think the biggest adoption has been for cattle producers because they're turning it into pounds of beef.
Yeah.
I don't know how much adoption we really have for truly people that are, grain only. I don't know that anybody knows that number, but it's got to be pretty small.
Okay. Yeah. Makes sense. Next one. Are you at the point that you can empirically assess how synthetic fertilizers might impact the soil health metrics such as gram positive?
I couldn't. I wish my coworker was sitting right here. But we get that question usually more frequently with respect to earthworms, and I have stood in a field that gets anhydrous ammonia and dug up earthworms. So I know that soils are a little bit more resilient than we give them credit for sometimes. So anhydrous ammonia being one that just a notoriously, hard on soil microbes substance, right? But you will still see them. There are still things there. So no, I don't think I could answer the question for gram negative quite yet, but but I can write that one down. I'm going to ask my coworker on that one.
Maybe a next one. Is anyone trying to add AMF to crops or cover crops? They've heard of this being
Oh.
done in prairie restoration efforts.
Uh, I'm sure there's a lot of different biologicals that people are trying out there right now in various ways, shapes, and forms. So yes, the answer would be yes, people are. But what is more impressive is the fact that if you just give the soils the right system, those things come back. They proliferate and they come back. So I think, I think it's more about the system than it is about adding a microbe, in my opinion.
Next one, what drives the persistence of continuous wheat and continuous tillage? I guess both there.
That is a good question. I think it is complicated. So I would say this, that if you are a continuous wheat farmer, you may not own a combine. So the wheat, the wheat harvest as it comes from Texas to Montana and into Canada can pick you up and harvest your wheat and you don't own a combine. I think that's one possibility. And I think another possibility is off-farm jobs for insurance, and things like that. Like it's a very simple system. So yeah, it's two different systems. There's almost three different systems. There's the livestock people that have a different sort of choices. There's grain only people, and then there's wheat continuous tillage people. And you might say, well, what about continuous no-till wheat? That's a horrible idea. That's never going to happen. That would have just a gazillion diseases and all kinds of pathogens. So never going to happen. So that's why the two go together. People that do plant continuous wheat have a lot of tillage in their system to combat that. In fact, we used to get distracted just watching the fires as people would set fire to the residue and burn the wheat straw off and then go do tillage. So, yeah.
They would burn their wheat straw. It's like not even bale it or.
Well, there is a lot of baling too, but there is still an awful lot of fires.
Wow.
So, yeah, there's people that'll bale that wheat straw. But there's a lot of 4.5 pHs in that area too. So, with sky high phosphorus. So, it's a very fascinating area.
Yeah.
Also, somebody sitting here would probably say, well, and there's also different populations too. So, yeah, I think it's really interesting.
All right. I think that gets to most of the questions we've gotten. And DeAnn, I want to thank you so much for an excellent way to wrap up 2024 for the webinars, for the Iowa Learning Farms webinars. This was really interesting. Again, if folks need a CCA CEU, please email Elena Whitaker, ALENAW at iastate.edu by 5 o'clock today with your name, the name you entered to watch the webinar, and your CCA number. Please start tuning in 2025 for the Iowa Learning Farms webinars, and also consider the perennial ground cover webinar series that's coming up in 2025 as well. There'll be a series of 5 of those on Tuesdays. With that, DeAnn, I'll give you the last word if you have any last comments.
I would just say thank you so much. It was fun to talk to some Iowans and beyond, so email me, call me, find me on Twitter. Happy to chat with you longer.
I was going to say she's a good follow on Twitter. So definitely, if you're on that platform, definitely follow her. So
we'll have to go post some more now.
Okay. Yeah, I know. I haven't done much for a long time either. So, all right. Thank you so much.
Bye, guys.